This is what is known as a karyotype. A karyotype is a tool that is used for genetic analysis. Using this mapping, we can detect if an individual has a normal set of chromosomes or if there's an abnormality. As you can see here, a karyotype is done on a diploid cell that contains 46 chromosomes. Let's test our knowledge. Why do chromosomes coil during mitosis? Select your answer, and when you are ready, hit play to check your answer. Good job! Chromosomes have to coil so that they don't become entangled while moving apart to the opposite poles. Think of it as if you had 46 balls of yarn that were totally undone, and tried pulling apart all 46 of those without making a giant mess or breaking some strands. As I mentioned a few minutes ago, a diploid cell contains 46 chromosomes. These 46 chromosomes contain the entire genome of an individual. This is how DNA testing can be done on a small sample of cells. These cells are regular cells in the body, such as liver cells or skin cells. In contrast, adults of a species have a special type of cells called gametes. These are the sex cells and are haploid, meaning they have half the genetic information of a somatic cell. In humans, these sex cells are eggs in women and sperm in males. The halving of genetic information makes sense because if a sperm and an egg both contained 46 chromosomes, the offspring would have 92 chromosomes. The duplication in chromosomes would almost certainly cause fetal death. Here you can see a visual representation of a haploid or a gamete cell, as well as a diploid or a somatic cell. As you can see in a diploid cell, there are 2 of each chromosome. In this example, the organism would have 3 pairs, or 6 chromosomes. Conversely, in this example, the haploid or sex cells only contain 3 individual chromosomes that when combined with another sex cell will create a new individual that is diploid. This image shows us a cycle of sex cells. Diploid individuals produce haploid cells that when combined create a new diploid individual. Take a moment to draw this figure in your own way to help highlight how diploid and haploid cells occur within the life cycle of a species. This will be your first figure to bring to class. Include in your personal figure a description of what type of cell is represented. And your textbook refers to other types of sexual reproduction cycles, but for us, we're only going to focus on mammal reproduction cycles. There's more to a cell life cycle than just mitosis. In fact, mitosis is only a small portion of the entire life cycle of a cell. Cells will spend a majority of their life cycle in interphase. During interphase, the cell will grow and prepare for division by increasing cytoplasm, dividing organelles, and in the S phase specifically, replicating DNA. We'll talk about DNA replication in Chapter 16. Make sure you have a firm understanding of how the mitotic phase that we will be studying shortly fits into the greater scheme of cell division. Using this information, try to work through these next 2 questions. I don't expect you to get them perfectly right, but try your best and bring them to class for points in discussion. We are now going to start looking at mitosis and meiosis individually. But first, what are mitosis and meiosis in the simplest of terms? They are cell division. While the products are different, they both essentially are just cell division. But first, let's have a joke. This image shows us the main differences between the 2 types of cell division. If you can get this image down, you'll be a long way towards understanding these processes and our objectives for these 2 chapters. This is your next drawing assignment. Draw this figure and bring it to class for points in review. Now let's look at the process of mitosis.